If you'd like to ask a question today, please use the Q&A box at the bottom of your screen. It will look like this. Our presenter does have another obligation at 1 o'clock, so we're going to do our best to get through all the questions with the time that we have with her. We're planning on ending at about 12:55. So today's presenter is Dr. Sarah Orlofske. Sarah is the curator of animal parasites and an associate professor in the biology department at UW Stevens Point, and also an associate editor of the Journal of Parasitology. Sarah grew up in southeastern Wisconsin and was always fascinated by aquatic life from a young age. She received her Bachelor of Science degree in biology and wildlife from UWSP and a PhD in ecology and evolutionary biology from the University of Colorado at Boulder. She's passionate about studying the multifaceted ways that parasites influence communities and ecosystems. Sarah, thanks so much for teaching us about parasites today, and I'm going to hand the mic over to you.
Thank you so much, and thank everyone for coming today. I'm really excited to share some aquatic parasites with you all. I'm a little bit biased in terms of my taxonomic flavor of parasites, but I'm happy to answer more questions about other types of parasites if those questions come along. And certainly the folks organizing the meeting are happy to jump in with questions. I think it would be better to kind of talk through any questions throughout the presentation rather than have you be confused as we go along. So thank you, Paul, for the introduction. I really appreciate that. I just wanted to include this slide with a little background on sort of my research interests. I've always been fascinated with parasites and diseases and how they interact with ecosystems, and particularly aquatic ecosystems, which is why I'm so excited to talk to a group that's also interested in aquatic ecosystems, lakes and rivers and wetlands. And you can see just a few of the species that I interact with here, and one of my favorite ways to interact with them. I love being behind a microscope, but even more so, I love being in waders and going out and playing with these critters. So I'm gonna get started with the slideshow. So I'm gonna start off with a little bit of basic refresher, or maybe an introduction to what parasites are and how we talk about them and what kinds of things parasites are and what they do. And then I'm going to dive into what I hope is a highlight for the folks in the audience, is talk about some specific examples, maybe things that you've seen or experienced and you want to know a little bit more about. And then I'm going to talk a little bit about our specific research questions in my lab and how we address some of the big picture roles that parasites play, particularly in food webs. We think about parasites taking, their host as a meal, but really parasites can also be a meal, and that might be a little bit different way of thinking about their roles and interactions. At the end, I'm going to try to save some time so that we can play with some live worms. So I do have some live parasites, and I'm here in my lab. So the not so exciting background is because I'm here with my microscope. There it is. That was a little bit corny, but we'll have more jokes throughout. And if you guys have requests for things that you want to see, I do have slides from our museum collection. As Paul mentioned, I'm a curator of parasitology, and we have about 22,000 specimens in our museum. So I brought out some of the real life examples of some of the parasites I'm going to talk about today. So on this slide, I show this when I start off my parasitology class, because parasitology is a really diverse discipline. All of the things on this slide are considered parasites, and this is what makes parasites fascinating as well as challenging to study. It's not a taxonomic group like, say, all of the fishes or all of the birds. These species are not actually closely related to each other. We've got crustaceans, we've got insects, we've got arachnids, we've got protozoans, some leeches on there. But all of these things are united by their life history. So how they interact with the environment, and particularly how they feed. So this is a little bit analogous if you want to think about studying everything that flies, right? If you studied everything that flies, you'd have to study insects, and you'd have to study birds, and you'd have to study bats, even though they're not closely related. So Maybe the first thing that jumps into your head when you hear the word parasite isn't the same thing that someone else has, but you're both equally right. So all of these things are parasites. So another thing that I want to just kind of plant the seed for is that there's no one feature that makes parasites parasites. There's not a single thing that we can point to say you are a parasite because of X. And I really love this definition from Brooks and McLennan that says that the only unambiguous definition is that parasites are those organisms studied by people who call themselves parasitologists. So I guess you have to take my word for it as a member of the American Society of Parasitologists. But in seriousness, the best definition we have for what a parasite is, and it's a lengthy one, is an organism that lives in or on another living organism, which we call the host, obtaining from it part or all of its nutrition or needs of existence and imposing a net detrimental effect on the host. So you can see it's kind of wordy, but the essential parts are that we have a interaction between two species that is a dependent one. The parasite needs the host and it needs it for either its nutrition or some other aspect of its existence. And the result of that interaction is negative for the partner, right? The host has some sort of harm. Sometimes that harm is self-imposed by the host's immune response, or sometimes it's a deliberate act of the parasite to in the process of taking its nutrition or other needs. So hopefully this at least provides a foundation for understanding the kinds of organisms and interactions we're talking about. One of the main sorts of reasons I love talking with groups like this is because parasites are everywhere and in practically everything. So I love this quote as well, that states that all living species are involved in parasitism either as parasites or hosts. We really hope that we aren't hosts, and hopefully those of you in the audience have never experienced being a host, but I imagine in a group this large, someone has had swimmer's itch, and we're going to talk about swimmer's itch here in a little bit. In terms of appreciating parasite abundance and diversity, in our ecosystems, each organism is likely host to many different kinds of parasites. The main hosts that I work on now are waterfowl, and we typically find 5 or 6 different kinds, different species of parasites, and upwards of hundreds of individual parasites in a single host. So it's pretty easy to understand that parasites outnumber hosts. And based on scientific evidence, about 50% of all living plants and animals are parasitic at some stage of their life cycle. So it's pretty hard to imagine an environment without parasites and how environments would be different if parasites weren't there. I think many of us kind of just take it for granted that parasites are there. They're small, they're invisible, they're inside their hosts for most of their life. So it's easy to overlook them. When really they're there, they're diverse, and they're abundant. So one other useful framework of just kind of setting the stage for understanding parasites and how they interact with their hosts is the idea of symbiosis. And this might be familiar to many of you in the audience when you think about other types of interactions, and I'll mention those in a second. But the big umbrella concept of symbiosis refers to 2 or more different species living in a close association. We typically think about this as a physiological or physical association that is long-term and carried out throughout an organism's life. So I apologize for the ugly Venn diagram. This is probably the most common Venn diagram that I use, but it gets at the idea that these are the main types of symbiosis. So we have things like mutualism, if you're familiar with plant and pollinator interactions, things like bees and wasps, and even birds and bats that serve a role for plants. And that's a beneficial interaction between 2 organisms. So both of the participants benefit. Commensalism is where one benefits and the other is neither harmed nor benefited, so it's kind of a neutral interaction. And parasitism is again, the negative positive interaction, negative for the host and positive for the parasite. And really the aim of this diagram is to illustrate that sometimes parasites don't necessarily have to be bad. If a host is well fed, if it has plenty of resources, if its immune response is not overreacting, a parasite might actually benefit its host by some other means, perhaps stimulating that immune response to not negatively affect other parasites or not negatively affect itself, as we've seen with human autoimmune diseases. So we can think about parasites essentially as symbiotic organisms, but also as a trophic interaction, so a feeding interaction. So this is how parasitism differs from a predator, right? Predators don't exist in a very long-term relationship with their prey. The amount of time a predator and a prey are both alive together is hopefully pretty short for the benefit of the prey organism. So parasites are feeding on their hosts, but they do so in a very different way than a predator does. So I'm going to pause here to see if there's any questions about the basics, about how we define parasitism or what parasites are before I launch into the more detailed info.
We don't have any questions in the chat yet, but we can pause for a couple seconds to see if any come in if you like.
Okay, well, as those questions come up, please feel free to put those in the chat and I'll try to keep an eye on that. So here's some of the nitty-gritty details, and this is what got me interested in parasitology to begin with. I was talking with Paul before we got started that I was really interested in herpetology as an undergraduate. I was part of the UWSP Herpetology Society. So I was really excited to learn about amphibian metamorphosis and how organisms change in their life cycle. And then I was introduced to parasites that do a lot of metamorphosis. So maybe that's a helpful way to think about a diagram like this. We can relate to seeing an egg, an amphibian egg in a pond, and how it hatches into a tadpole. And for a short time, it has external gills, and then those get covered up. And then it, does its growing and developing. It gets a big tail, it grows in size, and then it starts to develop legs. We see the back legs first, and eventually the front legs will pop out, and then it'll go through an awkward stage where it has a tail and legs until the tail's reabsorbed, and then it will emerge from the pond as a tiny metamorph froglet before it goes into the terrestrial ecosystem to further grow and develop before reaching sexual maturity. So the parasite's life cycle is a little bit analogous to that. It's going through different stages, and at each of those stages, it's doing something different with its environment and with the different hosts. So I'm going to talk mainly about flatworms because they're very visible and they're very important to aquatic ecosystems. These are the dominant sort of animal parasites in aquatic ecosystems. So the bias is kind of justified by their abundance and diversity. And so here you'll see that this diagram is set up with the bird at the top, sort of the 12 noon position. And that's because we consider the vertebrate host, in this case represented by the bird, as the definitive or final host, because that's where the parasite reproduces sexually. So that's where the adult parasites are going to mate and lay eggs. So in this viewpoint, we're trying to see how the parasite gets back to the bird through each of these transitions. So most of our worms are going to be in our vertebrate host, and they're going to be mating, and the result of that mating will be eggs. And those eggs are going to be released into the environment, typically through the feces, but sometimes through urine. But they end up in the aquatic environment. And that's the case for most of our flatworm life cycles. There are some significant and important terrestrial life cycles, but for the most part, our flatworms are tied to water, just like our amphibians are. That egg is going to hatch into a free living stage that looks a little bit like a paramecium, if you've ever seen a little tiny protist in the water. These guys have about a 12-hour lifespan, and their goal is to infect a snail. And this is why snails are super important in flatworm life cycles. That's the first stop on their journey. And they're going to reproduce asexually inside of that snail. So they're basically cloning themselves. So they're all genetically identical individuals. And through the process of developing those asexual stages, they will eventually form a tailed free living stage that leaves the snail and swims around in the water. And that's why these parasites are so important in aquatic ecosystems. So if we kind of go through this life cycle, we've started with the egg leaving the bird. It hatches into a free living stage. I'm just going to ignore most of the technical terms here. If you want to know more about that, I can answer questions at the end. But just understanding the transitions is probably the main goal. So that stage is going to infect the snail. Inside the snail, inside the reproductive organs of the snail, the parasite's going to reproduce asexually through a couple of different stages and then create those tailed free living stages. These stages actually kind of look like microscopic tadpoles, and those are the stages that I'm hoping to show you guys at the end. So if anyone here has unfortunately been a victim of swimmer's itch, that's the stage that you've been infected by. So if we follow our life cycle here, you can see a whole bunch of arrows come off of that stage. So I'm going to focus on this stage because it's really the essential one in terms of its interaction with the environment. So some of these stages can, in certain species like swimmer's itch, infect the host directly. So we can see that arrow going directly to the bird. The parasite's got chemosensory ability, so it can smell basically the host. It's also attracted to light, or it has gravitational cues that tell it where to be in the water. And it's going to essentially, like a torpedo, kind of zero in on a warm moving host, which it hopes is a bird, but sometimes ends up being a human. Humans are an accidental host for swimmer's itch here in North America. So that's the direct path. There are a couple indirect paths that these parasites have. One is to insist on vegetation and create a basically little capsule around themselves and wait until they get eaten. So this is the deer liver fluke life cycle. So if you're a hunter or just aware of deer flukes in the population, that's how deer get infected, by eating vegetation from marshy areas that have those insisted stages on them. So we've got two of our pathways kind of talked about, and then we'll move on to the most common way, and that is these little tailed free living stages get infecting another host. So this can be just about anything. This can be a crayfish, it can be another fish, it can be another snail, it can be an amphibian. Really, the sky's the limit for the types of hosts that can be infected at that stage. And now the shortest distance to that final host is for that second host to be eaten. So that line that goes from our small fish to our bird is consumption. And that's why parasites are so important in the food web, as I'll talk about later. There's one other little bit longer pathway that also involves consumption, and that is where our small fish is eaten by a bigger fish, or it could be a fox, or it could be some other vertebrate. And this is a transport host. So these hosts aren't necessary for the life cycle. They're not, they're more optional. And it, in some cases, helps the parasite move in the food web because a teeny weeny tiny little fish may not be as common a prey item for a particular vertebrate host. So these arrows are also consumption. So hopefully by kind of working through this general life cycle, you can appreciate some of the diversity of the more common and exciting parasites that I'm going to reveal here that hopefully are some that interests you in the group.
And we do have a couple questions here,
Oh,
Sarah, before you move
excellent.
on. With
Yeah.
that last image that you're showing with the intermediate host of the mollusk, are the parasites consumed by snails and other mollusks?
Oh, that is a great question. So there are two ways that the parasite can get into a snail host. One is by sort of torpedoing themselves in, sort of a direct penetration. Another is being eaten. So it depends on the species of parasite, whether or not it has one of those routes. Snails are really the only mollusk that serves as the first intermediate host for these flatworms. There's one kind of weird exception that uses a fingernail clam, but for the most part, it's always snails. Clams and other bivalves and other mollusks can serve as second intermediate hosts, and they're really good at that because they're filter feeding. They actually pick up the parasites through their feeding behavior, but they serve more as what's the small fish in this life cycle than the snails. So it's interesting because mollusks can turn up in multiple places in the life cycle.
And we have a couple more questions on this. People are loving this talk. What percent, you mentioned that the 50% of organisms are parasitic at some point in their life. What percent of organisms are parasites for their entire life?
Oh, that is a great question. I'm gonna have to get back to you with a solid number. There's some disagreements because we don't have all species described, and parasites actually lag behind free-living organisms in the number of species described. So we're underestimating the number of parasites when we make those kinds of conclusions. So I don't have a solid number off the top of my head, but I can tell you it's highly debated.
And then with the images of the intermediate hosts, are those negatively impacted like the definitive host maybe?
Oh, that's a fabulous question. That is an excellent question. So it's a little counterintuitive, but the final host, the vertebrate definitive host, actually has very mild pathology, if anything. So here in this diagram, the great blue heron is probably the best off of the bunch. The worms are very small. They have very fast metabolism, so they actually don't need that much energy. So that's one of the reasons why birds can be so heavily infected and have really no visible impact. So of this life cycle, the bird is probably the best off at being infected. The snail, because it infects, the parasite infects the reproductive organs, the snail is castrated. The snail will never lay eggs again. They're essentially little zombie snails that, snail themselves around and look like a snail, but they're really genetically reproducing parasites rather than snails. So they're essentially completely taken out of the gene pool by the parasite, even though they're still alive. The second intermediate host actually has the worst pathology because it's expendable. The parasite wants that host to be eaten, so it's going to do the most damage to the second intermediate host. And that's where we see probably one of the most fascinating parts of parasite biology, and that's parasite behavior modification. So in some cases, these fish commit suicide because their brains are infected by the worms. And they swim up at the top of the water and are easy pickings for birds. So they actually do things that are completely deleterious to their own survival because the parasite is manipulating them in order to be eaten. So if you had to choose a place in this life cycle, choose the final host and not the intermediate host.
Great. And then we had a couple swimmer's itch questions too, but you said you're going to touch on that later.
Yep,
Is
I'm
that
going to
right?
touch on swimmer's itch here in a minute. So maybe we'll hold on to those. And when we get there, if I don't address them, we can kind of pop back over to swimmer's itch.
Perfect. Thank you.
All right. I love the questions and thank you guys for those. Hopefully that'll help as we move along. So I work primarily in cattail marshes, sort of wetlandy habitats. So my background is in these habitats, but you can think about your favorite lake or your favorite water body as we go through some of the sort of cast of characters in this habitat. So I'm going to talk first about a parasite that's near and dear to my heart. So I worked on this worm for my graduate research. This is Riberoia ondantre. And you can see the adult worm that would be in the bird. In this picture here, we stain them red to help us see their different organs and identify them. So this worm is about maybe a centimeter long, and it lives in the esophagus and the stomach of the bird. But what it's famous for, and then as I was alluding to a little bit earlier, it's really bad to be an intermediate host. The parasite tends to be pretty ruthless. So you'll see this is that same life cycle, just a subset of those steps where we have the adult worm in the bird, it's laying eggs, those stages hatch out of the eggs, go into the snail gets castrated and then replicates those swimming stages. But here, instead of a fish, those stages infect amphibians, and they infect amphibian larvae as they're developing their legs. And as a result of infection with the parasite, they cause the frog to grow extra legs. So this caused a sensation back in the early 1990s when ponds in eastern US and places like Vermont, places like Minnesota, and then the West Coast in California were seeing huge outbreaks, 90% of the frogs having these limb malformations. And it was determined that in some cases, the parasite is responsible for this. And the thought is that this is an evolutionary adaptation by the parasite to be consumed by the appropriate bird final host. So the details of this life cycle, this has now become kind of the lab rat flatworm for a lot of disease ecologists for understanding parasites in aquatic ecosystems. This parasite is found here in Wisconsin. We found it most recently at Mead Wildlife Area, but it doesn't seem to be causing malformations in the amphibians. We don't tend to see malformed amphibians at all in our surveys, but this parasite is local, it is here, so it could be familiar to some of you. One of the other very charismatic flatworms, and for those of you who are anglers or interested in fish biology, this is something you've likely noticed because it's the biggest and most spectacular worm we have. Again, we have that bird as the final host, and then the eggs released, snails infected. And in this case, the larval parasites in that second intermediate host use a fish and become what's called locally yellow grub. So if you've seen yellow grub in a fish, you can see in this image, there are these little yellow spots in the muscle tissue. Those are the actual larval stage of the parasites, but this life cycle is really cool because it's a precocial development of the parasite. That parasite is as large as an adult worm in that fish host, which is why it's so visible and obvious to fishermen and women interacting with fish out in the field. You'll see a fish infected with this and be like, how are you even surviving? So we actually have to stain and mount the worms and actually check them for eggs. You can see the eggs in the middle of the worm here. That's really the only way to tell apart the larval stages from the adults, that knowing what host you got it from. So this is a big problem in like fisheries management and fish hatcheries because you'll have snails in these systems and then birds in these systems, and it will lead to high infection rates of a parasite like this one. So that's a very common fish parasite. And then here's the kind of heavy hitter in terms of the least popular parasite I can talk about. So this is the CDC's life cycle for cercarial dermatitis, which is the medical term for swimmer's itch. And this life cycle is a little bit different than the ones that I have been talking about so far, in that this is the one that causes direct transmission. So from the snail directly to the bird. There is no intermediate host. There's nothing eaten in this life cycle. So it's a little bit different. So you can see there's a mallard here representing our bird. Those adult worms live in the blood vessels, which is also a little bit different. Most worms live in the digestive tract or other organs, but these worms live in the blood vessels. They actually have separate sexes. So this is, again, kind of weird in the flatworm world that they're separate males and females. They have to find each other in the host and mate. They mate for life, and they live for as long as the host will live. In humans, they can live in the human form, the related species that lives in humans can live for decades. So chances are the bird's infected for its whole life with these worms. Eggs come out, infect the snail, and then it goes right back to the bird. What happens is humans get in the way, and because these parasites have such a short lifespan, they've got to find a host within about 24 hours or they run out of energy and die, that they come into contact with humans and then fail at infecting, but our immune system overreacts to them, and that's what causes the pathology. So if any of you have experienced swimmer's itch, this is a pretty mild case, and in some cases it can be overtaking, all your extremities, but this is the product of that larval stage called the cercaria, hence the name cercaral dermatitis, and it encounters and infects the human skin, and then those larval stages of the parasite provoke this immune response, and it gets worse every time you get exposed. So you build up worse and worse pathology, just like you would to a bee sting or poison ivy because of that allergic reaction. The parasites are killed, so it's not like the parasites are actually infecting you. They die in the skin. The immune response is capable of killing them, but it's all of the breakdown products of the parasite that stimulate that overactive immune response. So I think that was my brief introduction to swimmer's itch, but let's see if I answered those questions or if we still have some more swimmer's itch.
Yeah, you definitely answered one about how they get into humans in the first place. We had another one here, though, asking, why are some water bodies more prone to infecting people with swimmer's itch than others?
That is a great question and a very active area of research. So I could probably give a whole presentation on swimmer's itch. But one of the things that really determines the swimmer's itch probability is the snails and the snail habitat and how numerous the snails are and how well they're infected with other things besides the swimmer's itch. So in a very diverse parasite community, you'll actually have snails infected with other things. The stages within the snail for swimmer's itch are actually pretty wimpy, and other parasites can outcompete them, so they actually fight over the snail. So in certain places where you just have a very simple snail community and you have one snail that's the right host, that can lead to higher incidence. So having more different kinds of snails and having more different kinds of parasites seems a little counterintuitive. We think more parasites is a bad thing, but in this case, more parasites, swimmer's itch is a really bad competitor as far as parasites go. So it actually takes a pretty depauperate community. The other thing is swimmer's itch parasites are really poor swimmers. So if you saw the flyer, that little worm that was in the picture that looks like an anchor, it has little kind of bifurcated tail, like a little dolphin tail. It actually hangs up at the top of the water because it can't swim very well. So when wind directions blow the waves in towards the shore where people are swimming or recreating, that can drive infection. You'll think, oh, there's no swimmer's itch here or this year or this season, and then all of a sudden you'll get a wind and it'll come from the right direction and it'll push those parasites into where they're interacting with people. So there's different drivers. There's the snail population, and then the parasite community, and then also the environment that can kind of work together to create sort of hot spots and cold spots of swimmer's itch incidents.
Thank you. And then we're also wondering if you have any prevention tips on swimmer's itch. So things like toweling off quick, or would sunscreen be a preventative?
The best way to prevent infection is to get out of the water periodically and vigorously rub yourself dry with a towel. Air drying is really bad. That gives your parasites a chance to penetrate. So if you can at least remember to periodically, maybe every 20, 30 minutes, get out and really vigorously rub yourself off, the parasites don't infect immediately. It's not like a bacteria or virus. It takes a while. So as long as you're pretty diligent, that'll reduce the ability of the parasite to get into your skin. There's not really great like over-the-counter sort of creams or lotions or things to prevent it, but just being aware of wind direction and the presence of snails in those habitats. Maybe, okay, the wind is blowing towards the shore, a bunch of algae and things are being moved this direction, maybe not a great day to be out there, and just be mindful of, getting out and toweling off is going to help.
And can you see those or other stages of the flatworms or parasites with the naked eye, or do they need to be seen under a microscope?
That's a great question, and something that one of the reasons I brought our little worms in today is they are visible to the naked eye, but given any type of turbidity or any other organic matter or other microorganisms in the water, you're not going to really be able to tell them apart. So what we do is we isolate our snails in snail jails. So we put them in 50 mil centrifuge tubes, and that's how we're able to kind of hold these up to the light and extract the worms from the tubes. So you could take a water sample and see them, but you'd have to know what you're looking for and be able to tell them apart from other little protists and amoebas and ciliates that are in the water as well. There are methods that were developed in Africa for human schistosomes, so human infecting blood parasites, but that requires DNA analysis and filtration and sort of more sophisticated techniques.
Great. It sounds like we're going to have you back for Swimmers Itch Talk because there's a lot of questions, but we have some general ones too, but I'd like to see what else you have in your presentation here before we get too hung up.
And I can also brush up on some of the newer literature with Swimmers Itch too. It's not something we encounter a whole lot because no one wants to swim at Mead. We don't actually see a whole lot of Swimmers Itch in our snails. I've probably only seen 2 Swimmers Itch snails in my entire career, but we don't work in the types of areas where Swimmers Itch is common. So it definitely pops up in different places. So we'll leave Swimmers Itch behind. And I have just a couple other sort of rogues gallery parasites to kind of intrigue you by. This is near and dear to my heart. We work on waterfowl, and these are the bad guys. And I wanted to talk a little bit about the bad guys. So these are introduced parasites. So some of you might be familiar with the faucet snail. Faucet snails are an invasive species of snail. They're very small. They're very cryptic, and they are unfortunately part of the life cycle of 3 different parasites that also came with them over in their migration from Europe. What's unique about these 3 worms and why they were successful at invading along with their host is that the snail serves as both the hosts in the life cycle, so both first and second intermediate hosts. So it really is a 3-host life cycle, but only needs 2 different species. It needs a snail and it needs a duck. And the ducks are getting infected by eating them. And when the ducks eat them, some species, particularly American coots and lesser scaup, die. It's a very deleterious infection. It affects their intestinal tract and they die from internal bleeding and hemorrhaging as a result. So we're monitoring where the faucet snails are and what birds are infected with them and where this could potentially be an issue. So in some of the bigger lakes, Lake Michigan, Lake Winnebago, these are common, and it's an interesting dynamic to keep track of and to watch for the health of these birds. So my last one, as I kind of try to cover all the bases here, is a very fishy one as well. So a little bit different than yellow grub that we saw earlier. This is called black spot, and it gets its name black spot because of the black spots. So you can see here, this fish is really heavily infected. This would be pretty rare in nature. Some of the worst infections we actually see in sort of commercial environments, again, because they kind of build up infection from birds and snails and fish being in the same place. But these parasites are in the genus Diplostomum. I've tried to avoid scientific names throughout, but there's really not a good common name for these guys. This is a diverse cosmopolitan group that's economically important because it affects fish. We're most familiar with the black spot like you would see here. But there's also a really cool set of species that infect the fish eyes and cause blindness in the fish, and that also requires them to swim up higher in the water and become easily preyed upon by their birds. So very cool worms that do deleterious things to their fish. And I'm not going to spend too much time in the weeds here of this evolutionary tree, but I did want to point out a couple of lines on this tree which represent different evolutionary lineages, because this is actually a new species that we're working on right now from Wisconsin that is in otters. So we don't know what the fish host is. We don't know where the larval stages are. We found it as an adult worm in the otters. But hopefully once we get a handle on what this is, we can then figure out what fish these otters are eating, where they're picking it up, how widespread it is, if it causes any problems to the fish, or if it's just one of any number of culprits for black spot disease in fish. So
does that happen to be the same thing as ichthyotherias?
So it is a different thing than that. There's different symptoms of black spot. So this one is related to the flatworm infection. There's other types of infections that cause black spots, but this is just one specific cause of it. Yeah. All right, I can tell I'm talking too much. So let me just quick hit on the last little bit of this, and that's kind of why we do what we do in my lab. So many of you are probably familiar with food webs and how we look at primary production in our plants or algae, how that moves up through herbivores and carnivores to some of your top predators. And you can see a little aquatic food web here in the corner. So the goal of a food web is just an ecological map of who eats who, how many species are present, how many interactions they're taking place, how long are those interactions, so how long is a food chain inside, and how strong are those connections? How many different things eat the same prey? How many different predators eat that same prey? Because that can tell us who might be vulnerable to extinction or what secondary extinctions might happen in an ecosystem if we were to lose a species. So it's not just a cute diagram in a textbook. They're actually really useful and an active area of scientific research. So what we do in my lab is we put parasites in them. The parasites are in the ecosystem. Hopefully I've made the case there that we do see them. We've just kind of ignored them because they're small and they're inside a host and they're difficult to identify. But I wanted to show these diagrams. These are 3D food webs, and you can see in the first panel, the green are the primary producers and the red are the free living organisms. And then in panel B, you can see the blue are the parasites added in and how many more organisms we can now account for and how many more interactions. And then panel C has the same number of species, but includes this principle of concomitant predation, which is when parasites get eaten along with their hosts, right? The great blue heron is not going to pick out the yellow grubs from its fish. It's going to swallow the whole thing. So it's going to eat those parasites along with the prey. So that bottom panel is the most realistic depiction of what's going on in our ecosystem. And what I worked on for my dissertation research and what I still work on with my research students here is how those little cercaria stages that cause all those problems, right, with swimmer's itch, how they're actually eaten. They're prey in the ecosystem, so those free living stages are consumed by a variety of aquatic vertebrates and invertebrates. I actually love aquatic macroinvertebrates, so please come to Jessica's talk in January. She's going to talk about some of those little creatures. So some of your dragonflies and damselflies actually eat parasites, and they're super important for the ecosystem for a number of reasons, but eating parasites might be another one. So last but not least, I think this is my last slide here with some data. This is work from my graduate work in California. So we're still working on a similar approach here in Wisconsin. But you might be familiar with some of the different taxonomic groups on the x-axis here. So we've got amphibians, we've got gastropods, which are the snails, the beetles, your odonates, your dragonflies, and damselflies. And this represents 3 different ponds that we were working on, and the biomass of these different groups of organisms that we know play really important roles in the ecosystem. So we can see kind of this dashed line is the mean trematode biomass. So all of the parasites I've been talking about today are trematodes or flatworms. And you can see that even though they're invisible to us, we don't see them in the water, we don't see them inside their hosts, they have as much biomass as your dragonflies and your true bugs and your beetles. So they're probably playing a pretty significant role in that ecosystem, even though we don't see them. So this is pretty eye-opening. We wouldn't ignore those groups, but we tend to ignore the parasites, but hopefully knowing that they're there and the roles they're playing will help us better understand these aquatic environments. So I'm just going to rush through this real quick because I can show you how we actually look for these parasites in the environment. We put them in tubes. We go out in the field, we dip net these guys out, and we isolate them in the tubes, and then we put them under the microscope. So that's all that I have in terms of prepared slides. I'm happy to answer questions, and I'm also happy to put some of the little worms up on the screen if you guys want to take a look at them.
Yeah, I definitely want to see. I mean, I'm not that I speak for everyone, but I think it would be very neat for everyone to see some worms under the microscope.
Yeah, so I can answer questions while I'm kind of getting these in a dish.
Does that mean you want to answer questions off camera, Sarah?
Oh yeah, I can. Sorry, I had to get a little bowl to put my worms in. But I can answer questions while I put these on the scope for you.
Okay. We do still have a lot of questions about swimmer's itch, and I'm curious if you have a good source for more information that might be like maybe most encompassing of swimmer's itch information.
Off the top of my head, there are some new published papers on this topic. So I think it really depends on sort of which aspects folks are the most interested in, if it's a prevention method or if it's a control method. There's not sort of one resource, but I can compile some of the newest research that's been done. And sort of share that with you if you want to share that out with folks who are interested along with the talk. I'm not sure if that's possible to post that where you post the recording, but I can definitely do that.
Yep, we can certainly share information with our registered participants.
Perfect.
We did have a question that was upvoted as well, wondering if you consume a fish with parasites in it, are those parasites killed by proper cooking, or is it still a risk for us to become infected with them?
Absolutely. You can properly cook your fish. Cooking will kill the parasites. Freezing may not. So some of the parasites can withstand freezing temperatures. So it would depend on how cold it gets and for how long. It would be required to be frozen, but cooking will kill the parasites. Salting, smoking, and drying will not. So you can still be infected by parasites if it's dried fish or smoked fish or pickled fish. Cooking is really the only surefire way to make sure you are parasite free.
If you did consume a parasite by not, properly cooking a fish, what would determine if that parasite is if it survives or is digested?
So that's a great question. So many of the parasites here in North America, like black spot or yellow grub, those are bird parasites. So it's very unlikely that those would make a jump to mammals. But in places like Southeast Asia, those parasites normally infect humans. So those you would want to be particularly wary of. So lots of folks ask me if I eat sushi, and I don't. But that's not because I'm a parasitologist. It's because I didn't like it when I was a kid, and I just never grew up to really appreciate it. So it really depends on sort of what type of fish, what type of environment it's in. In terms of probably the biggest risk of infection is actually some of your salmonid fishes in lakes in Canada that have a tapeworm. I didn't talk about tapeworms today, but Diphyllobothrium is the broadfish tapeworm, and folks get that from eating salmonid fishes. So it really depends on the particular type of fish and the type of parasite that you're potentially encountering. Got some worms here for you. Now, can you guys see the screen with the microscope camera, or did I lose my screen sharing?
It looks great.
Yeah, we see it.
Okay, so let me just get a little bit more zoomed in here. So what you're going to see, these are the Cercaria stages of the parasite in the water. You're going to see them just kind of zooming around, right? So that's them. Let me get the white balance on here. Here. Oh, no, that is not good. There we go. That's a little bit better lighting. But there was a question about, can you see them in the water? So this is about 1 millimeter. That's the scale bar. So you're going to see these guys shooting around on the scope. This particular species is phototactic. So they're going to try to swim towards the outside of the dish where the brightest light is. So that's them swimming around. So if you get infected with swimmer's itch, the worm that causes swimmer's itch is about half that size. So this is a big one. This is a big chunky worm compared to swimmer's itch. I'm going to see if I can get one more close up for you guys. Take a look at.
You want to take another question while you do that?
Sure.
Have there been any studies about the impacts of climate change on parasite populations?
Absolutely. And the answer is we don't know because it affects every life stage differently. It affects different hosts differently. So we are making strides to understand how climate change will affect parasites, but because there's so many different, there's about 40,000 species of flatworms, which if you think about it, there's about 10,000 species of birds. So there's about, 4 times as many parasites of flatworms as there are birds. So it's hard to say exactly what's going to happen in every case. But we do know that with snails, and snails being super important, as snail life cycles become faster and you have more abundant snail populations, that's likely going to lead to an increase of your flatworm population. So that's sort of the one kind of direction that we have. But in terms of what happens, sometimes the parasites are more sensitive than their host to temperature. Sometimes they're less. Things like drought will really be hard because if snail populations get knocked down due to drought, that can have a negative effect on them as well. Let's see if I can find a little guy on the slide for you. There we go. So he's going to be moving. I'm going to switch cameras here. There he is. And you can see how active they are. That little tail is their battery pack. So this guy, he's going to swim like this for about 24 hours and trying to infect a host. This species is one that infects amphibians. This guy's going to try and find a tadpole and swim in their kidneys and then wait for that tadpole to be eaten by any number of birds, really. We find this guy in ducks, but in great blue herons. Basically anything that eats a tadpole is going to have one of these worms. Where'd it go? Got away from me. Oh, he escaped. He's out from the cover slip. He is making a break for it. You can see how fast they are. This is what my students do in the lab all day. We chase these guys around on the screen and try and identify them.
He's desperately looking for a host.
He's desperately looking for a host. And what's interesting about swimmer's itch is if you put your thumbprint on the slide, if you just give it a little bit of a chemical cue, it will try to actually penetrate the slide. It will be convinced that it smells like a potential host. So it'll actually drop its tail like a rocket booster and try and infect it. It's kind of neat to manipulate them that way. So I lost him.
Sarah, we have all kinds of questions for you. I feel like we could do a whole webinar just with the questions that we have right now. But realizing that you have a 1 o'clock meeting, I want to give you the opportunity to say, I need a couple minutes to go get on this other meeting, or do you want to take one more question? Or
let's go ahead and take one more question, and then I'm happy for you to share my email, or I can come back and kind of do a swimmer's itch particular. That'll give me a little more time to gather some more resources on that topic, and then we can go from there.
Okay, I did take a screenshot of all the questions as well, so I could email all of those to you if you want to
fantastic.
Write down some answers. We could share those back out as well. Emily, maybe if you're okay with it, I'll just pick one that's up on my screen right now. It's referring to the larval phase of native mussels and their impact on the native hosts, on different fishes and things that they infect. Can you talk a little bit about that?
So really, I'm not the fish parasite expert, but I work with a lot of fish parasite experts. So I'd be happy to kind of touch base with some of the folks who work on fish parasites in particular. My understanding is it really depends on the intensity of infection. So hosts that are really highly infected, they have numerous parasites. So the more parasites you have, the worse the pathology. So it's not like having a cold or a flu where you're just like, you've got a cold or you've got a flu. With these types of parasites, you're really sicker the more of them you have. So it happens to be how many parasites are infecting and also what life stage. If you're a very small individual, a very early life stage, and you have a lot of parasites, that's going to be very detrimental versus you're a mature adult and you're getting infected later in life. So I hope that that's at least helpful at this point, because those are two of the most kind of general concepts that kind of guide what kind of effect those parasites have on their hosts.
Okay, wonderful. We have lots of comments that said, this is awesome. This is such a cool webinar. So people love this.
Well, thank you. And thank you so much for having me. I really appreciate your time and sharing this opportunity to talk to people about something that, you know, it is important, but maybe goes unnoticed. So I'm glad to see that there's so many people who appreciate it.
Absolutely. Well, we're going to cut it off there. Thanks again, Sarah, for spending an hour with us. And thanks for all of you for attending. We'll hope to see you back here on January 14th.